when I first applied for funding to research wild bees. That was 2015-16. I was told they don't exist, we're not funding you to study them. So it's taken me about 4 years to get funding, so we had to go out with no resources, no staff, no nothing, and just try our best to prove that they existed. We wouldn't be able to do any of it without significant support from beekeepers around the place and also members of the public. So just to kind of a little bit of an introduction anyway, first is that honeybees actually, it's one bee but two very different lives, and beekeepers sometimes don't think about this. Honeybees were first and foremost a wild species that humans have tried to manage. Officially they can be described as domesticated, but they're not domesticated animals. We can't control what they eat, we don't control how they mate, the vast majority of us. So they're not domesticated. They're managed or semi-managed is about all we could actually apply to them. However, beekeepers have had, beekeepers often talk about the custodians of honeybees and they're saviours of honeybees, but actually beekeepers have a massive impact on that wild species, which in my view, we have a responsibility to think about when we're beekeeping. So as well as, trying to improve local bees and trying to get the best out of our bees, we also have to have an ear and an eye to the wild members of that cohort, which actually, we wouldn't have probably the diversity and the strength of our managed colonies only for the wild population. So, and also now more and more beekeepers are actually using wild colonies, learning from wild colonies, and they can benefit us even if we never capture a swarm and bring it into your apiary. The health of your colonies are partly due to the health of the surrounding population. So wild honeybees first do exist. So this was, up to fairly recently, nobody was sure whether they did exist or not. And actually they don't exist across a large proportion of Europe because of humans. 2014, the IUCN Red List has, and that's the current red list for Apis mellifera, is down as data deficient because the panel that reviewed the current status of Apis mellifera as a species could not determine whether any wild versions of that species existed anymore. Because actually in a lot of European countries, it only exists really in the managed population. There was some studies by Moritz done a number of years ago, and there was a paper by Jaffedal in 2010 which showed actually in Europe, in the countries they looked at using a genotyping approach, most countries in Europe, say for example Germany, the genetic diversity could only be explained by the managed population, whereas in Ireland they detected that we were likely to have wild populations because the genetic diversity couldn't be explained just by the number of managed bees. But even when I started, so I'm relatively new to the beekeeping research actually. I only started really around 2014. Once I started going to meetings and stuff, I first started hearing reports from beekeepers about the existence of bees living in other places apart from hives. We sampled the first colonies in 2015. We look at these terms, feral, free living and wild. So the one study I'm very familiar with in the UK, it talks about feral bees. We've proposed the use of the word free living bees in our 2020 paper because these are bees that are living outside hives. We don't know if there have been escapes from colonies or when they might have escaped, or I'm sure the direction has gone back and forward a few different times. Some people would actually suggest that we use the term wild anyway, if they're living outside of an apiary, that they're living in the wild. Either way, whether you're talking about them being feral, free living or wild, they're still really important. They're living in wild conditions for a period of time, and lots of people in their managed apiaries cannot keep their bees alive without chemically treating them. So these populations, be they feral, free living or wild, are important for biodiversity and for beekeeping. Tomorrow I talk more about the first study we did from 2016 to 2018. And that was really going off half-cocked, trying to just find bees and so on. Then we because of the vagaries of research in one person in the country, in Ireland, doing this really, we partnered with the National Biodiversity Data Centre to try and accumulate solid data from around the country so that even if I stopped working on bees, that the Native Irish Honeybee Society or FIBCA, for example, could take over this work. And this is an online reporting tool for members of the public to report the existence of wild colonies around Ireland. And we have reported up nearly 500 instances of colonies living in a variety of places right across the country. And this map is from 2019. We have a lot more since then. Ireland has fewer trees even than the UK, so bees have adapted to humans. So most of our bees are actually in cavities and walls, the roof spaces, and they do live in trees as well. But we've cut down most of our old trees, and we don't manage our forestry very well sometimes. So bees have adapted to human habitation, and a lot of our reports are centred around where people are, because people are noticing them. So we have a body of work to do to look at our national parks and forests to look there for evidence of bees. That takes time. The bee hunting method of Tom Seeley and so on takes a lot of time, and we haven't had funding to do that, but we have funding now, so we'll do it next summer. These are just some pictures. That's Chiara Benetti, master's student from Italy that came over last year to look at it. We developed a bee hunting method with Joe Ibbotson and Philippe Salvini here in the UK, who were working on the bees in Blenheim and Boughton Estates, to help look at, we have so many forests and woodlands, and the bee lining method of Tom Seeley is for really a very localised area, and we're looking at much bigger geographical area. So you're looking at broader features to identify honeybee nests. So these are just two examples of trees that we found, and one key on the right-hand side, you can see the tree there with the fungus. The fungal shelf is a key indicator of decay inside that tree that would provide a good cavity for bees. So obviously buildings are a big location. So our castles, and we have a lot of castles and the like in Ireland of various condition, and a lot of them harbour bees in the walls and cavities. The top picture is a lodge gate of Lochoutra Castle near Gort in County Galway, and the bees are actually in the kind of roof wall space in the top of that building, and have been there for quite a number of years and are doing really well. The last picture of the tree on the right was a tree from this castle estate. And then we've got some houses, like the picture in the bottom left is a house in the middle of nowhere in East Clare. The bees got in there a number of years ago. The one in the middle is a tumble-down cottage. The arrow is pointing to a small hole, probably in the old chimney of that cottage. And those bees, I've been monitoring them personally the last few years and collecting swarms from them. And then the one on the far right is a church. So churches as well, particularly way up high, they're very hard to sample. But there are a lot of bees in our churches. This map is from this year, my student sampling this year, and Chiara last year. We have now sampled Donegal very intensively. So Donegal is the top northwest corner of Ireland, and actually they didn't have varroa for a long time until some Egypt from Dublin brought bees with bugfast bees with varroa into that area. But anyway, they're doing a really huge amount of work. Boomtree Mick, I call him, is a Dutch forester living in Donegal. People thought he was a bit weird for a long time, but now they've come around to his way of thinking, and he's got beekeepers in Donegal behind him, and they are trying to undo the impact of people bringing in non-native bees. He's carved and put out about 200 log hives. He puts them out, they get occupied naturally, and they leave them wild. So there's lots of projects. This is just another project in Ireland that I know of, and we're collaborating with Mick now to help provide genetic data for him. To support his work on requeening, for example. They're in the UK as well. These are just two people I'm working with at the moment. Joe Ibbotson and Philippe Salvini. So Joe is working on in the Boughton estate, and Philippe has been looking at the bees in Blenheim estates. You probably all have heard that nonsense in the media last year, where people, got some items of clothing in a twist. About this work without really seeing the benefit of the fact that in each of these places there are about 40 colonies that are living year on year without human intervention. Some of the traits are like what Tom Seeley would have described, but some of them are quite different. These are some of the oldest oak forests in Europe. There are amazing cavities, and these guys are doing amazing work. So expect to hear some cool stuff coming out of here in the future. So this is other example. Roger has mentioned yesterday. He's a load of experience of looking at and having to remove wild colonies all around the UK. And the bottom are pictures from Blenheim, looking at some of those old trees occupied by wild bees. Free Living Bees is a website and a project run by Jonathan Powell here in the UK, really looking at, and Matt Somerville is another person who's been putting out log hives here in the UK, similar to what's happening in Donegal. and they're doing a load of work on wild bees. This is a website from a guy in Portugal who has gone into this movement as well with log hives and studying wild bees in Portugal. And Elise Pinto, a collaborator, is now that she's been alerted to this, she's now coming across wild living colonies in Portugal. So there's quite a large number of people observing, monitoring, trying to capture or protect honeybees that are living in the wild. So we can and we are now making moves to change that IUCN input. Instead of being data deficient, we now know there's forests in Germany and in other places and in Poland where they're finding bees living in trees. So as these people are observing the bees, they notice a lot of things about the behaviour of the bees and tricks and tools to maybe modify their own beekeeping practice in terms of cavity or cluster size, honeybee size and colour, swarming behaviours, nest locations, and so on. Much of the data, though, is not collated. So these guys are going out doing this work themselves. It's not published, so it's missed by mainstream science and mainstream beekeeping. And I'm hoping that this will come in. You'll start reading more and more about it in the coming years. So these pictures here are just some taken by Joe Ibbotson, who has noticed that on any year when he opens, is investigating cavities, that if some of the hybridised bees are much more unhealthy than the ones that are darker. And in fact, a lot of the colonies that end up in the wild become darker over time. And I think, Roger, you would have noticed the same thing, that we see very few queens headed by non-native bees surviving very well. They end up becoming, what we say, more mellifera than mellifera themselves. We have a saying in Ireland, I don't know if you know that invaders become more Irish than the Irish themselves, and it's similar here. So Honeybee Watch is an international group that's been put together really to try and accumulate information, not just on wild bees, but also on managed untreated bees. There's a larger movement now as well, realising that maybe we don't need to treat our bees quite so much. So if you around the room are familiar with, I know some people have seen wild colonies, it would be amazing if you could just Google Honeybee Watch and log the occurrence of these wild bees. There is a move to maybe have the UK as a pilot study for Honeybee Watch to try and launch a coordinated effort to look at wild bees. So the question is, of course, well, can they survive? Yeah. We see, there is a school of thought, skepticism says that, well, these wild bees, they're escapes from local colonies, they're going to die out, they don't persist. So that's an obvious question that we have to prove. So this is a paper just from this year that was sent to me actually, and they have been finding wild colonies now in Germany. It's interesting, when I went to Finland at a SICO meeting a number of years ago, probably 6-7 years ago now, I was surprised to hear that beekeepers were told to kill colonies that they found in the wild. So no wonder they're not surviving, because of the perceived threat of wild colonies to beekeeping. Anyway, in Germany, obviously they've replaced their native bees with Carnica by and large, but there is a movement there to, allow native bees. And so it was nice to see the discovery of wild bees. This paper, though, are saying that like 90% of the bees died out each year. So they survived the summer, but they're not surviving that first winter, and kind of dismissed then the idea of these bees being a sustaining population, because 90% of the bees were dying out and they were replaced by swarms from the local beekeepers the very next summer, and that there's this pattern of replacement. But 10% of the bees that are there are surviving. So what about them? And I don't think the work isn't old enough to find out actually if this is the case and what happens to those 10%, because Tom Seeley in his work would suggest that yes, a high proportion of newly established colonies will die out, because obviously a lot of the ones that might escape from local beekeepers are probably not that robust and not that fit, and you would expect that a high proportion would die out maybe in winter and not have the fitness levels of the traits to survive winter, particularly I guess if they're not native. But Tom Seeley found that yes, 23% of founder colonies perished over the winter, but actually those that established had a survival of 84%. In Ireland, of the ones we monitored 76 colonies from 2000. The first ones were 2015, they died, but really from 16 onwards. 14 of those colonies are still going strong. We've sampled them a number of times and we're monitoring them. So in Ireland, we're fairly confident that these colonies are living beyond the time at which you would expect them to die due to varroa. That graph is a little bit misleading there because it suggests that only 2 colonies survive more than 4 years. A proportion of those bees that were documented, that was kind of a cutoff of whatever, 2018, and at that point, those colonies had been alive for those periods of time. But some of those colonies that were alive for 2.5 years are still alive today. So not all of them, it doesn't mean that they died after that time point. It just means at that time point they were living for that length of time. So we have at least 14 colonies of the 76 that are still going now. And so we're now continuing to monitor the 500 or so colonies that we have reported since that time. So we can't confirm that lineage has persisted in all of those colonies because they're being monitored a couple of times a year. They're all over the country. We tend to ring the people after the summer to see if they've survived, and then we ring them about March and see if the bees are still active, have they come through the winter. And with some of them we talk more than others. It's hard to keep track of everybody. But there is a period of time. We know spring is quite tough in Ireland quite a lot, and so they could be there in March, but they might not actually have survived till May, till the swarming period. So we're improving that. However, using genomics, now when we first sampled the bees, the first time around, we didn't sample a huge number of bees. And so now we're using a genomic approach to look at colony diversity. And when we compare, so some of the ones we have, there are 2 examples where we have bees from 2016 and 2020, where we've had enough bees to do a colony level genomic approach, so 30 bees. And we can see that those colonies are more related to each other than anything else, even very local bees. That's from an area in Spiddle in West Galway. They're in a roof space of these 2 old bachelors, and we've been in constant touch with those. So this is really good evidence that these bees are surviving in the roof spaces for that length of time. And we have another one, was in a roof space of a bungalow, and in that bungalow are living ecologists and an ornithologist who know about bees and ecology and can confirm that swarms left their roof, but no swarms came into their roof. We had to remove those bees recently because the roof was being destroyed, which is how the bees got in there in the first place. So just in case you're probably interested in Blenheim and the Boughton estate here, it just shows you that this is the Boughton estate here, the top ones, and they're grouping so this is a schematic diagram from genomic data, and it's really like a pedigree. So if you were to think about you and your siblings, your parents, your grandparents, being linked in a graph over time, this is the same thing using genomic data. So when bees are grouped together in these clusters, it indicates relatedness. So these are Carnica bees here as an outgroup, and the Blenheim bee, that sample, that one colony we tested is largely Carnica. The ones from the Boughton estate actually group with Apis mellifera, but there is some introgression from C lineage bees definitely, which is not surprising because it's in the UK, but fairly high amounts of mellifera. Just to say about Blenheim, this is just one sample. The sample size we got are very low because Philippe puts his hand into the colony and picks out bees, but we didn't have 30 bees. So that hybrid nature is not indicative of all the colonies, just the one we sampled. This is just data from morphometrics from those Blenheim and Broughton estate bees. You can see this one, it's consistent with the genomic data. The Blenheim estate colony there, the few colonies we did with morphometrics are largely hybridized. The Broughton estate ones, though, have large proportions of AMM present. So Joe would kind of observe that the bees become dark over time. I'm not saying black, I'm not saying colour is the be-all and end-all. Talk about that later if you want. So then we've kind of looking at genomic signatures of selection. So we really want to look to see, is there something different about these bees that can survive in the wild that beekeepers can use? Can we detect at a genomic level genes that are under selection, some behaviours that facilitate their survival? So we're right in the middle of this analysis, looking at the direction of gene flow when bees become hybridized. My hypothesis is that most gene flow goes from the local bees into anything that's brought into an area rather than the other way around. But the bees that are brought in can really disrupt the integrated genetics of local bees quite badly. We have got this funding. I've been able to hire Alex. So she actually gave a talk here last year. Alex did on the top. She was did her project with Stephen Martin, Professor Stephen Martin, and now she's with me. And then Arrigo Moro, who has been working with Peter Newman in Switzerland, and he's quite heavily involved in Honeybee Watch. He's now working in my labs for the last month. So together we'll be looking at what is the molecular basis for fitness adaptation to allow bees to survive without such beekeeper assistance. So first thing is to improve our monitoring and establish a monitoring protocol. So we want people to monitor 4 times a year to capture the after summer, directly after winter, spring, and after swarming. So we want to see that those bees are still there at that time, and we're developing a mobile app that people can go out and just answer very few questions. Fewer the questions, the better, the more responses we'll get. Alex had a look at some of our data for this year, and the point really was that you can see the preponderance of log hives with our recent research, and Mick has been doing really good monitoring of the log hives since he started putting them out. We have some tree cavities, as you can see, and tree cavities, we do have some tree cavities nests that are still there since 2017. So even though the cavity is really small, the persistence is still pretty good, but most of the persistence is in roof cavities, and so roof cavities provide a really good space for wild bees. We've bought some thermoimaging software to look at that in more detail, to see the differences in what is the nature of the cluster, how big is the cluster, and so on in that environment. Because the queen, I think she can leave the old comb and move on to new comb, nice and warm and dry, no problems with damp. We're looking at does management impact gut microbiome of honeybees, and our preliminary data suggests, which you can just see visually down here in this graph, if you look at just the colour, it's just a heat map, you can see there's some more red, I would say, to keep it simple, in the managed colonies than in the free-living colonies. But you see these different types of microbes appearing. So I don't have enough data yet. We're in the middle. My student is in Switzerland in the middle of analysing. It's taken us a long time. This data was impacted by the fact that we collected honeybees largely from the wild from the entrance, whereas the managed bees, we were taking them from the frame. So I did wonder whether we were sampling more nurse bees in the managed colonies in comparison to foragers in the wild. So we've gone back with all of our recent work, and we sampled the managed colonies in exactly the same way now to mitigate that potential conflict. So how local is local? So wild honeybees are going to be able to tell us a lot about this. So what is the direction of gene flow? It's clear from the data, I'll show more tomorrow, that basically the wild population is the same population as the managed. They're a free-living, free-mating, openly breeding population. But we definitely have evidence of local breeding going on here. So this really does add support to the evidence that's coming out about the importance of local. But we will be able to look at how local is that. Because here I'm just showing you, this is a cluster from Donegal that we've recently sampled, and there's a second cluster from Donegal, and I don't really understand that, why these are 2 different ones. Maybe this is originating from Dublin and that has been brought in, I don't know. But we also have separate clusters, Connemara, which is extreme west of Ireland. They have had a beekeeping association. They've been trying to keep things local. They've been trying to share amongst each other and breed only from local bees, and that's evidence in the genomics. Connemara bees are coming out as separate from everything else. Bees in County Louth are also distinct, and other bees like Cork and Kerry are mixing, but we definitely have, even the bees around me are distinct from a beekeeper about 6-7 miles away. So we're picking up signals of local mating, which is consistent with Cooper's work on UK and Irish bees. So in Ireland, what's the story? Most beekeepers in Ireland are hobbyist beekeepers. About 90% of them keep AMM. So these are results from a survey we carried out last year amongst Irish beekeepers. And actually most people in Ireland will breed locally. So breed using, the queens will mate near their own apiary, at their own apiary or locally. So we have this kind of interesting behaviour in Irish and UK native bees, where you have inbreeding and outbreeding exhibited by our bees, depending on the weather and climate at the time. So it allows our bees to cope with our inclement weather conditions. So we have very natural and specific brood patterns and cycles described in the grey literature. Some of it's dismissed, but actually a Norwegian study recently shown that there's very clear differences in the subspecies in reproductive behaviour. So I have asked a question here whether both the weather and how our bees operate in UK and Ireland could actually help them in their fight against Varroa. So again, more and more beekeepers in Ireland are not treating. So about 30% of beekeepers now that you talk to and reply to surveys like this are not treating. They might use some biological control, but they're not using chemicals. And we're starting to kind of build that community in Ireland. Recapping, our studies have suggested that recapping is rare, but in Ireland recapping is one behaviour you talked about. We're not finding it at all, really. A couple of bees across multiple apiaries, but grooming reports are very common in Ireland, so damage to bees, it seems to be the way. We have a research apiary, about 20 hives this year. A lot of them actually are stemming from wild colonies. So this one here was one I caught from a colony that had been living in a tree for quite a number of years, and it's my most prolific, healthiest, best colony. And I've generated lots of splits and started spreading this around. So these are 2 daughters from that colony. We have a swarm from one of the long-term colonies that's been alive since 2016. Your man gave me a swarm from that last year, which we're hoping to perpetuate and again breed from for beekeepers. And then we have a number of other colonies that have resulted from swarms coming into our apiary, because I find with bees there, it's very attractive for swarms. So I always leave a few hives in my apiary. I don't believe about disease. You can talk about that later. I'm not worried about that. And so they're quite good, strong colonies. I have a satellite apiary that allows me to make splits and control stuff from my other apiary. Small colonies, occupied bait boxes that we are really using to look at survival. We're doing minimally management, not feeding them, I have to control for swarming in a way. I have to do artificial swarms or something. I have to let the bees go through something, but I have to manage swarming because I'm at the edge of a city. So I don't want to be accused by Roger of bees and chimneys around the place. Which it was in my first year. I kept getting calls about in the city with swarms in the city. They were always blaming me. I don't think it was my fault. So this is just showing you some of the data from last year from our colonies, showing evidence of biting and damage. So you can see that Mull 1.2 there has got the highest amount of damaged, a good lot of mites on the bottom board, and a lot of them are damaged. And this is the colony from the wild colony. So it has got some good behaviours, lovely healthy bees. Some beekeepers came to do a thing at our apiary, and they were like, oh my God, these bees are just beautiful bees. Nice, black, shiny, productive, nice bees. They don't sting me either. CL1 is quite low. That is from a wild colony, but it had quite infestations of chalk brood. I've let that die out now this year. So we make a good bit of splits when the colonies get too big and whatever, we sometimes have to control it. So, and then normally my approach is to give these away for free to beekeepers on the understanding that they don't treat them and that they pass on that favour down the line to another beekeeper in the following year, that they make splits from them. Nice brood pattern, nice black healthy bees. Things won't improve until farmers stop using chemicals to control things as a first port of call, and this was his plea for improving things for insects. And you have to ask yourself, is this not the same for honeybees? Should we be preventatively treating our colonies as a first port of call of treating Varroa? Are we got too complacent and are chemicals just too easy? I hate them. I'm like Jeff, I'm lazy. But I don't need to treat, I've never treated. And my colonies are on the go since 2018. The most recent stock I've lost before that was due to incompetence as a new beekeeper, nothing else. So could an untreated, unmanaged bee population provide a source of Varroa-tolerant resistant bees for beekeepers? Well, there are examples, and Jeff talked about some of these yesterday. So Gotland in Sweden and the Arnott Forest in New York. Everybody knows Tom Seeley, everybody knows his work. So it is clear that the bees can live with Varroa. So Varroa are present, but the bees are tolerating Varroa. So I would say yes, we can do this, but they might not have all the traits desired by beekeepers. And another thing here, I guess, is that there might be an initial loss. So there has to be some cooperative bee breeding. That's something I'm trying to encourage in Ireland as well. We tried to have a breeding programme a number of years ago in Ireland to breed Varroa-resistant bees, and it failed. And it failed because beekeepers were working independently. And there's a risk to a beekeeper. We love our bees, right? You don't want your bees to die. So if there's any risk of losing your bees, you'll treat. So it would be better if beekeepers work together or associations work together so that you don't have to not treat all your bees, but that you think about before you treat. And you treat, after thinking whether your bees need treatment, or you work with your man down the road, and if you lose some bees, that you agree to make splits for each other or help each other out if you fail. So I do think it needs thought, it needs work, but I do think it's valuable to do. So we've got a difference between host tolerance and host resistance. And I think what we have in Ireland, at least, is host tolerance. So it's the ability to reduce the effect of the parasite. So the mites are present in our colonies, but they don't really have much damage to the colony, even if we don't really know the mechanism. Host resistance is really the ability to reduce the fitness, i.e. the ability to survive of the varroa, so to prevent really varroa building up in a colony. Well, the colonies we've looked at apiaries in Tipperary and in Louth and in Galway so far, and in none of them do we see recapping, and in all of them do they mention grooming. And John McMullen, who's done quite a lot of work in Dublin, would also probably agree with that. So can bees evolve faster than varroa? Yes, they can. Varroa are clonal, so they've got SIB mating, so they're all very genetically identical, whereas bees have a much higher rate of recombination. This is when bees are producing egg and sperm. The chromosomes from both parents in the queen, say, for example, line up together. They duplicate, and then they swap bits of information. The parts of the chromosome swap over each other, so it creates a lot of variation. And this is very common in honeybees. So honeybees can adapt quickly. Honeybees can change quite a lot, and certainly faster than varroa. There is a lot of evidence, just Google it. There's a lot of people doing this work now, mite-resistant traits being developed. So some different studies across the top. This is from a really nice paper from Locke in 2016 that's well worth reading. And it shows basically some of the traits that between hygienic behaviour, grooming behaviour, colony size is important, reduced development time, and reduced mite reproduction. And I do think in Ireland and England we have our weather to also help us in varroa resistance, because I'm not sure with the brood breaks and the reduction of laying in winter, not in winters like this when it's really warm, but anyway, most of the time it is conducive to low varroa. I know there are exceptions. So natural selection you might be aware of, which is an ability of populations to adapt. So individuals, in any population there's variation, and within that variation different individuals have a better propensity for survival and reproducing. So this is Darwinian fitness. Providing that variation is inherited, and we know a lot of the traits that allow bees to survive are inherited traits. And if you leave them alone, a gradual change will occur, and this is adaptation, right? So I would ask, well, what do you want from your bees? I would say the humans are driving selection of different traits in honeybees, which are at an artificial level, which messes with natural selection. So I would ask you, individually to think about what you want from your bees. Most beekeepers are hobbyists. They're interested in beekeeping for the bees and for biodiversity. And they like gardening, and they want to do something with some land or their time. So really, why buy bees in from abroad or from somebody who imports them when you can get good local bees that are adapted to your own local environment? So that's the first question. Second question is, why use nasty chemicals on your bees to prevent disease? I don't want to handle these chemicals for my own health and time, never mind for my bees' health. So if they're impacting varroa, you can be sure they're impacting the bees as well. So maybe it's not necessary to treat all your bees all of the time. That you think about this, do a sugar shake or an alcohol wash or bottom boards. We did bottom boards last year, and it was good. Twice a week we counted the bottom boards, and yes, there's a lot of skepticism out there, but you just common sense. That's the thing. It's not very common sometimes, but have a look at your bees. Are they healthy? Do you see lots of deformed wing? Do they look okay? And if they look okay, well, why give in to propaganda? Right? But I would hedge your bets also. So if you've got a nice colony and you're afraid, you have to mind your bees, but think about it. Why take away all their stores? So honey we love honey. It's brilliant medically for ourselves, and that provides this social immunity for honeybees. So why take off all of that good stuff from the bees and give them sugar water? Why would you do that? Yes, from an evolutionary point of view, bees overproduce, so maybe we select them to overproduce honey. So we can take some off, but leave them a super, leave them a lot of stuff in the brood box. Don't be taking everything and giving them sugar water to feed on. When we take that honey, because we sell it as an artisan product for a lot of money, because humans value it for their health, both for wound healing and other things, as well as our throats. It's the same with bees. If your bees are stressed, why have huge colonies? Why have large productive colonies? Why do you need it? Do you want all that honey? Like, what's the point? You have to lug all those boxes. The bees are under stress trying to sustain that colony. You've got a June gap, and next thing you have to go out with fondant because the bees can't feed themselves, they all starve. So why do that? Why get bees from somebody from outside that aren't fit, that aren't adapted to our local conditions, that can't adapt their brood rearing, can't adapt their use of forage when things get tough? You want tough bees. If you're a hobbyist and if you love bees, you want to think about some of these. And this is coming from wild bees, because they're surviving right beside you when yours mightn't be. And then why drive selection of such docile bees that they can't defend the nest? You want bees with a bit of character. You don't want them stinging you or driving you out to the field, but you want them to come up and say hello and ping you and nibble your hand. And, and if you squish them, you want them to sting you. Otherwise, how can they defend the nest, right? How can they bite varroa? How can they groom? How can they damage mites? How can they do that? So if they're very, very aggressive, you want to look and see and talk to somebody if you've got an F2 hybrid, right? You might want to requeen. But local bees, if they're happy bees, they're not at you. They just want to be left alone. And I would ask, actually, don't be going out every day or once a week to check your hives. We love our bees. It's addictive. It's an addictive activity. But you're stressing the bees every time you go out there. So, bear that in mind. So my point here is small-scale beekeepers are not bee farmers. That we're not. So we don't have to follow the same propaganda that's being taught, and use common sense. We don't have to follow the same methods that commercial beekeepers do. Commercial beekeepers are entitled to be commercial beekeepers, but most of us are hobbyists. We don't have to do the same thing. So what beekeepers can learn from wild bees as well: don't overcrowd your colonies in the apiary. This has been mentioned a few times. This is a lovely study by Seeley's team, 2014. Over here at the apiary, there's densely clustered beehives all in a line, and then he compared it with more spread out, more dispersed colonies, and found big differences in survival and mite counts. So there was much lower mite counts and lower swarming in the colonies that were dispersed than in the ones that weren't. And even in the colonies that did swarm in the crowded group, they developed high counts. Swarming was more frequent, and that swarming is a good way of managing mites. But even in the colonies that swarmed in the crowded group in the apiary, they developed high mite counts much later. So much lower mites, much higher survival in the dispersed colonies. So you can just do simple things to separate your colonies a little bit, have them in different directions, make sure it's, to limit drifting between your colonies. It will improve disease much better. So disease is much lower in wild colonies. This was a misconception out there for a long time, and it probably still is there. People are afraid to bring wild swarms into their apiaries because of disease. In actual fact, most wild colonies have less disease than managed colonies because there's much less horizontal disease transmission. Apply colour, different direction hives. Consider not having colonies too large. I talked about that. And consider your swarm control methods. We have to swarm control, but just think about the myriad of methods out there and see which one might be good for you and the bees. So to conclude, wild honeybee colonies are present and survive multiple years. Managed colonies are part of the same population as the wild population, so they could also survive if we let them survive. Honeybees are not domesticated, so we can never manage them as we would cattle or sheep. Most beekeepers are not commercial, so different approaches are valid for those of us who don't have huge numbers of colonies. Local breeding is definitely evident in our bees in England and the UK. Local adaptations are highly likely. Heard about someone yesterday, a student talked about different dialects in bee communication. I'm looking forward to hearing that research. So I would say respect this where you can, and actually also tell the beginners to get bees locally before they go online and buy bees. Commercial beekeepers, then, I would ask that they prevent the impact on other local bees, wild or managed bees locally in their local environment.
You talked about how local local is? And isn't there a kind of a big problem in how you define what is wild and what is managed? Because you have this feedback loop between swarms going out and living there, particularly if, as you've described, beekeepers are keeping local bees, which are better for the environment they're in, and then those are then swarming back from the wild into effectively the beekeeper's apiary, and how you get that categorization correct for scientific purposes.
I would love to see us, not everybody, but in pockets, if we accept that it's the same population, why get hung up on the pedantics of wild and managed when, if we look after the wild and we look after our managed and we know that they're breeding, then beekeepers can use the diversity in local bees, and if we're not importing, bringing bees from far away, then that will happen naturally. I don't really see a huge problem with it. There's a problem if, I guess, local beekeepers are treating chemically, so you're going to have this impact of artificial selection. So you're always going to get bees swarming into the local environment that are not fit necessarily for varroa because they've been treated. So I think in our managed colonies, if we're treating regularly, we're actually artificially selecting for weak bees in terms of varroa. They may not be weak bees in other traits that we want. And it's just something to think about. If people reduce treating, it's less of a problem.
In the wild colonies you've been able to observe for a number of years, how do they manage their own survival? So how do you have any data on how often they swarm or supersede or?
No, that's what we're trying to gather now in this number of years. Like the preliminary work we've done suggests that Nosema is quite low, Varroa is low. It depends, the small tree colonies, I think it depends on cavity size. So we've got a thermal imaging machine now to look at that. So we'll match different cavities, so trees and roof spaces, for example, with managed bees. So we now have different cohorts of fully managed, minimally managed bees in roof spaces and bees in tree cavities. So the smaller the cavity, the more they'll swarm. So we are seeing swarming and from beekeepers putting out bait boxes. So some of the ones I'm monitoring, they'll swarm a couple of times a year. So I put out bait boxes and they get filled. Others, like there was the one in the roof at the very start of the talk, for a number of years they wanted to get those bees out, and I was like, well, wait till it swarms, it'll be small, we'll go in, because we thought actually in that roof space it was going to be a massive colony, and it had been there for over 10 years. But in fact, it didn't swarm very often. I guess some years it swarmed twice, other years it didn't swarm at all. So, and the supersedure, it's hard to get into the brood. We can't get into a lot of these spaces, so their cavities are behind very small openings. So the MIC in Donegal has made us some log hives with the side entrance, so that we want to leave them established and then we'll be able to get in. We don't want to interrupt the bees too much, but from a science perspective, we'll be able to get in and look at brood. So we're going to have to look at that. I mean, the work by Cooper is suggesting about Irish and UK bees selecting, mating locally and selecting for supersedure. He suggested that those traits were recessive. So I'm going to try and find in our genomics, see if we can access. We have to try and use proteomics and others as well, but we're going to try and access that information. What are the genes that are under different selective pressure in these cohorts of bees? So I hope to come back to you in 5 years with some more information. But it's really hard to study the wild bees because they're inaccessible. So we're actually we have cohorts of bees, again from Donegal and Galway, from minimally managed and wild bees. We're sending them off for disease screening for viruses and foul brood and amoeba and stuff like that to a lab in Spain independently. To then we hope and be able to compare them. And we're comparing the microbiome of all of these as well to see what's going on.
I'm a beekeeper from Woodstock, so very close to Blenheim Palace. And I was very interested by the comment that the bees surrounding Blenheim Palace are buckfasts. And I was just wondering where that piece of information came from. I did have one buckfast colony a few 100 yards within the Blenheim estate, for about 4 weeks, about 18 months ago. And as far as I know, I've got 9 other colonies about half a mile away. None of them are buckfasts. I've got friends who have got colonies very close by. None of them are buckfasts. So I was interested to know where that information came from, and does it skew your information at all? Thank you.
Actually, I'm delighted to hear that. Absolutely delighted, because, so with Blenheim, I have very little information because the sampling, and you know, I do have a lot of pictures and images of the colonies, and a lot of them are dark, and some of them are banded. So it was really, I think, Philippe, in frustration, knowing about buckfast beekeepers coming in, and he's been trying to work with the Blenheim estate to actually limit that. So I guess we haven't mapped all the beekeepers around the estate. He's been putting up bait boxes around the perimeter to try and capture swarms coming in, and we are slowly processing them, and we're doing mitochondrial DNA analysis on all the colonies now, because at least we can tell the maternal. And I'm hoping that a high proportion of them will not be, and I'm really excited to hear that if there are dark bees up around Blenheim, that is really good, because that means I do think that the dark bees survive better, and that means that there's really Blenheim will be a real gold mine. So watch this space. We will be generating data. So thanks for that. That gives us hope, and I'll feed that back to Philippe.
Can you explain how importing bees undermines the integrity of the local population?
Basically, I guess we sometimes think when we hear about genotyping methods, we hear about mitochondrial DNA or this or that and the other, and we sometimes think about single genes, but actually evolution happens at the population level and to individuals. So we're not just talking about single genes, we're talking about, through evolution, the whole genome and the phenotype evolving over time. So what can happen when you have an import, so there can actually be a few things. One is that if imported bee comes in that is very different and has very different traits from the bee that's already there, so then the hybrid, the offspring which is hybridized, doesn't have the same traits as the original native bee in terms of that have been adapted to that environment. So it could have traits that, say we're talking about ligustica. We don't get much ligustica in Ireland, but I'm going to say for that sake. So you get ligustica coming into an AMM region and interbreeding. So basically some of the traits of the offspring will not be adapted to the local conditions. And so they've been adapting to living in the weather and climate in the UK and Ireland for thousands of years. And so somebody brings something in the last 100 years, it hasn't adapted. So Carnica, for example, and ligustica will be used to warm, dry conditions, and they're brought in here to wet weather. They can breed, but what is the phenotype of that hybrid? Is it better fit for warm conditions or wet, wild, windy weather here? So you have, sometimes of course the subspecies are the same species, so they can interbreed, but even so in honeybees, it has been remarkably adapted to very different environments across Europe. And I do think that if we left the bees alone, that some of those subspecies will become separate species. And I think Apis mellifera is really distinct, and it's got some really distinct traits in Northern Europe that have allowed it to adapt there, and it'll move further north as things change in weather. So a couple of things can happen when you hybridize. One is that you can get introduction of alleles that are of benefit. So I'm not going to say, some hybrids, you get hybrid fitness, and in the first year some hybrids are really productive. You get big hives that bring in loads of honey, and it's way hey. But if the genome of the incoming bee is very different from the incoming of the existing bee, sometimes you get what's called outbreeding depression, and it's equivalent almost to inbreeding depression, which we know is bad. So when things are too different, in the second generation, we know that when A. mellifera breeds with Carnica, we get aggression. And that's because the gene complexes at a genome level are so different that it causes these aggressiveness in bees. So like with scutellata going into the US, those Africanized killer bees, they're not killer bees. Some of them actually have conferred an advantage in the US, and they're being really successful. But they're limited in the north and southbound. Sorry, I've answered you very long. So what happens normally with migration is either new alleles come in that can confer an advantage or a disadvantage, or they can change the allele frequencies locally. And if the local bees have adapted to the situations locally, then bringing bees in from too far away, it can be a problem. It's happened with conservation of deer and horses when they try and boost a declining population, and they bring in examples of the horses from the same species but that have been in zoos or from quite a far geographical area. They bring them in, they mate, but the offspring, the deer or the horses that get of the hybrids, although it's the same species, they're not, the timing of the events are slightly skewed because of the different genomics in each side. So the foals are sometimes born at the wrong time of year, so they're not born at the right time for grass, for example. So that outbreeding depression, it causes a problem.
Hi, I think you said that you don't use the beelining technique. Could you say a bit more about the techniques you do use for finding wild colonies, or is it just you were waiting for them to be reported to you?
Actually, I'm hoping to put a talk. I've developed my website at the moment, and the master student Chiara has done a nice talk on that method, and we've put together a method, and I'm going to make it available on our website as soon as I can. But really, it's just putting together features of an environment that you might use to look for bees. So we were looking at basically satellite imagery of an area to look for local woodlands near gardens maybe or something would be the first thing. Then you're looking at forage availability in an area. You might look at whether there's gardens. I would look to see if there's tumble down buildings or what's in that area, what major landscape features. When you go there, you're looking for water, and you're looking at puddles, and you're looking to see are there bees, because they will use muddy puddles locally, right? Then you're looking at flight paths, and in the flight paths you're keeping an eye to see whether you see flying insects. So you're looking then in an area for suitable trees, so beech, ash, oak, old trees, or tumble down buildings. So these major kind of features we would look at in an area. We would assess an area, think, oh yeah, that has potential, we go there, and we walk around and we look at all of these other features. And the first picture there, the tree and the girl climbing the tree, we both came to that tree independently using that method. It's not perfect, it still takes time, but it's just, in your local area you could walk around and look at kind of what would bees like here, and I bet you'll find bees.
There's been some research done in Poland about probiotics in bees, quite successful using conventional types of probiotics you might use in human foods. And the research group there found that there were some specific bacteria, new species which have been identified in honeybees, which were capable of degrading polyfructose type molecules and fermenting fructose directly. So this is an epigenetic type of phenomenon which can rapidly diffuse through different colonies just by transfer of the gut microflora from one colony of bees or one bee to another. Do you think that's something that could be significant?
So my understanding of epigenetics is more that it's not a transfer of actual bacteria, but more gene expression. So it's more, so whether the probiotics are switching on and off genes, is that what you mean?
Just the fact that they are present in the population, and so you have some groups of bees which can ferment fructose and fructooligosaccharides, and others can't. And so these bees are actually more resistant to Varroa.
So with the microbiome data published until quite recently, there was thought to be a fairly depauperate microbiome in honeybees and a global one. So the same species found throughout the world. And we did some work in Ireland to study this, because I find that really hard to believe, because if you think of how differently bees have adapted to different environments, I would believe what you're saying. So we did a study where we combined looking at pollen and the impact of pollen on microbiome. But our microbiome study at the time only used the standard method, which was a very small portion of a gene that's present in all organisms. And we didn't find anything different. But I really believe that we must have different strains or different species around the world, and they just haven't been picked up yet. And my student at the moment is just about to finish. We've used a pool-seek approach, so we've combined 30 bees from our colonies, and we're up to nearly 200 colonies where we have this data from now, and we have included abdominal tissue. So most studies take it from the thoracic muscle, which wouldn't have microbiome. We've actually included some from the abdomen. So I'm really convinced we're going to find some novelty here in the bees, and it will be interesting to see the impact of management or wild on those, and whether we have new strains in Ireland, for example. We have to have some, because where the forage is different, if they're foraging for thousands of years on different plants than in the US, there's bound to be new microbiomes. So I'm not surprised, and I hope I'll be able to report back in a couple of years to say, or you'll see a paper coming out to say, yeah, we have. So I'm not surprised at all.
This group actually did the original work with lactobacilli from probiotics from Hansen's, as it happens, for human milk, and they found with that they got increased varroa resistance just by feeding these compared with feeding water to control.
That's cool. Yeah, so we did, the one I showed there, you couldn't read the names, but we had there's 3 or 4 different species of lactobacillus in our data so far, of which some I haven't heard of. And they're differentially distributed in the colonies. They're not in every colony. So I will now go back with a renewed vigor to look at our data. Yeah, that's great. Imagine, because we all know the holobiont is proposed, so important for human health. It's the same with bees, and we know very little about it. So we do need to look at that, and I do wonder what the dynamics are. And I'm sure it must help in disease free or disease-resistant bees. It would be lovely if we could show that these bees that have never been treated have a different microflora to the bees that you're treating with chemicals. And that would be further encouragement, perhaps, if we can show that.
I'd like to share that I've been involved with a project in Somerset, and in 2017 I did a bee audit on an estate, and it's actually a visitor attraction. So it was really important. They wanted to have bees, but it was really important to me because my apiary is nearby that we didn't introduce any bees. So when I started, we did the bee lining, did a bee audit, and we found 2 wild colonies in trees. We now have 7 trees across the estate that bees live in from time to time, and they're not all oak trees. We've got ash, pine, a lime tree as well, and a horse chestnut. So it's really interesting, and we observe all our colonies every week.
Wow.
And we've gone from the 2 colonies in 2017, and we now have between 15 and 20 colonies, and we're not losing any in the winter. We're not doing any treatment, we're not feeding any sugar, and we only harvest honey up until midsummer. So we're finding increased amount of propolis, so the bees are really defending themselves well against wasps and predators. We've even noticed propolis curtains on the inside of hives that come across. And our queens, the colonies that we do manage in more conventional hives on the estate, we find that they're really dark, and we've got really dark bees, and we're observing them flying at 5 degrees in the winter. So we're pretty sure we've got Apis mellifera.
So would you be able to input that information of those into Honeybee Watch?
I'd love to, yes.
That would be, because we're considering using the UK as a pilot. So, I've been doing work in Ireland for a while, so we have it started, and no need to waste it on Ireland, but we're looking at trying to find funding to kickstart a real study in the UK as a pilot for this. We're gathering data. It's open, the portal is open to put in data, information there in Honeybee Watch, and then hopefully we will. But even if you want those genotyped, send me samples. I need 30, and we'll genotype them and have a look and see.
That's
what you
brilliant.
want. Yeah, but I think it's really important that news gets out there, and that
Yeah. I suppose the next thing is, or my question is, how do we communicate with the local beekeeping groups about this real importance of not bringing in more bees? Because in the West Country, thousands of bees are brought in, and it's, it is part of the culture, and it's very divisive in the beekeeping community.
People are entitled to bring in bees. I think I think a lot of beekeepers are quietly doing stuff, and they're nearly afraid to say it because the convention is X, Y, Z, right? And I think, there are more and more people beekeeping for different reasons, So I think one of the challenges in Ireland as well is beginners. If we don't have enough native bees for beginners, or we don't tell them enough in all our introductory class, at least we should have a lecture on local bees, right? There should be one lecture in every course on local bees, just to raise, people can do what they want, but just to raise the idea, hey, we have them. But also I think associations need to work better together and actually have bees share the information and try and rear queens for beginners. I think that's really important, and for each other, that there's, use the associations, use your own bees, but share.
Yeah, I think that's really great.
We're not all after money. Like, fine, some people have to make a living out of it. We don't want to interrupt or interfere with people who have to make a living out of selling queens. But there isn't enough of a market anyway, so we're not going to impact that. So a little bit of cooperative beekeeping, I think, will go a long way towards that. And write for your local beekeeping magazine, and go on Twitter, and go have a website and have a blog, and just, get it out there.
I do quite a lot of work at Oxfordshire Beekeepers Association apiary, and they're all buckfast. And no matter what I say, they bring in buckfast bees all the time. So, and it's quite close to Blenheim, unfortunately. So I think we need to get the message out much more to local associations, because once you've got the senior people in associations buying them, then the beginners are going to buy them.
Yeah. I mean, I think it's worth noting that Buckfast Abbey have stopped generating buckfast bees for quite a long time now. They don't believe in buckfast bees. I think everybody knows that Brother Adam unfortunately was wrong in a lot of what he said, and with the best will in the world maybe. So Buckfast Abbey are using local bees.
I've come across a tree which has some bees there, and I think they're wild. You said honey bee watch, and after that, what do I do?
So if you go onto Honeybee Watch, there is a portal where you can put information on about that hive, about those colonies, and that's the first step. We're trying to increase the reports of wild bees so people will believe they exist. That's the first step, and know they exist. And then, as I said, we're desperate at the moment. We're meeting every week trying to find avenues. We have a list of philanthropy, a list of companies, a list of grant agencies that we're trying to get money for a UK pilot, where we will develop a monitoring program of these bees officially, and sampling, and kind of do what I'm doing in Ireland, but in the UK as a cooperative thing.
Right, I'm going to finish off with a question, please, if you don't mind.
I do mind.
Before this lecture, somebody downstairs said to me they enjoy the banter that's going on here. Well, over the years, there's been a lot of banter between Grace.
He thinks it's banter.
Well, most of it seems to be at my expense anyway. The question I was going to ask was, on your map, you showed one dot right out on a peninsula on the southwest corner. Would you expect to find different results in that particular colony than any of the others, because it might have been isolated?
Ideally, yes, but we know that area, they import bees quite a lot. So that's the Kerry Peninsula, and in Kerry and Cork, they bring in a lot of buckfast as well. But actually what is beautiful about that area is that it's so mountainous and tough that the buckfast seem to be corralled in one kind of valley. And the buckfast people there and the Apis mellifera people there are very respectful of each other. So if there's a beginner in a mellifera area, the buckfast people won't sell to them, and if there's a mellifera, if somebody in the buckfast area is looking for mellifera, they won't sell either. So the buckfast seems to be distinct. So we know that in Ireland in the 1920s, after Isle of Wight disease, there was huge imports of bees from the Netherlands into Ireland. Not huge huge imports at the time, like now. There were skeps of bees. So in Kerry, I know there's a number of different circulating strains of AMM, and people are moving bees. So it's not actually in Donegal. So the bees from Kerry and Cork seem to be kind of mixed up. It'll take us a while to find if there's a little pool, like we have to look at the forest. Maybe if there's an isolated forest in that isolated peninsula, we'll find new genotypes. We're still after to see, you know, the question before about wild managed, I'd still love to find, we'll do some dating work on our genome, and it would be lovely to find some old genotypes, and that might be a good place to find an old genotype, as well as Northwest Donegal.